Thank you, George, for the introduction. Again, my name is Josh Neufeld, and I'm an associate professor in the Department of Biology here at the University of Waterloo. And also, as George mentioned, I'm a microbiologist. I'm a microbial ecologist. So I understand and I study how microorganisms interact with each other in the environment out there, but I also understand how they interact with the environment of us, the human body. So I sort of have two roles tonight. The first is to provide very brief context for everything we're going to talk about. And then secondly, to moderate, to introduce the speakers, and then to field questions from you for those speakers that will eventually become the panel that will be sitting up on the stage again. So for context, I too want to welcome you to the University of Waterloo. And I am very pleased to see that so many of you could make it, that so many of you are here healthy tonight, because each one of you has come here and sits in your seat with 30 trillion human cells, but you've come with friends. You are also joined by 100 trillion microorganisms, the bacteria, the archaea, and the fungi. And with these organisms, I've not even mentioned the viruses, probably outnumber these other microorganisms. With these organisms, you coexist in peace, in harmony. They do you no harm. In fact, they probably help you. They probably train your immune system to fight infection. And they probably benefit you in ways that we're only now just beginning to understand. So I thought it would be important to provide that context, because we're about to talk about a particular pathogen. There aren't very many pathogens relative to all the microorganisms out there, but as we begin to talk about this pathogen of primary global concern at the moment, I want to make sure that we don't paint with that same broad brush all the other microorganisms that are here with us this evening, which do us, again, no harm. So we are now going to begin talking about Ebola, global concern. And we're going to be listening to presentations by 3 faculty members here at the University of Waterloo. As George mentioned, this is a partnership between 3 different faculties. And the complementary expertise that you'll be hearing from include what is Ebola, and then how does Ebola spread, and how can Ebola be treated? Those are the 3 talks you'll be hearing, and we won't be stopping in between for questions. Hold those questions, keep them in mind. We will come back at the end when the speakers are then invited to come up on the stage, and we'll then take questions and discuss Ebola in more detail with you. Okay, so that's the way it's going to work. If you're okay with that, then I'm going to be introducing our first speaker for this evening. And I want to make sure that I introduce them correctly, so I have a bio here for you for our first speaker. And so our first speaker is Dr. Christine Dupont, who received her PhD from Massey University in New Zealand. Her research focused on the isolation of bacterial proteins as vaccine candidates for sheep and cows, which you may know are the most important citizens of New Zealand. I'm reading, these are not my words. Christine returned to Canada for postdoctoral studies at the Ontario Veterinary College at the University of Guelph, where she turned her attention to similar vaccine work with horses. This ended up being a short engagement because she soon joined us at the University of Waterloo as our first lecturer in the Department of Biology. Christine teaches several undergraduate courses in the areas of genetics, biotechnology, and microbiology. And by the way, her Master of Science degree is in virology from the University of Guelph, and Christine still recalls her former professors talking about the mysterious early Ebola outbreaks. Somewhere in there, she also obtained a Bachelor of Education degree from the University of Windsor. And so, with that, would you please join me in welcoming Christine Dupont.
Thank you, Josh. It's so wonderful to be able to connect with the local community here tonight. I thought a good place to start would be with what is a virus? Viruses are genetic entities, life forms if you like, and they're composed of a genome that's made up of either RNA or DNA, and that's wrapped up in a protein shell. And for a lot of viruses, that's it. For others, like Ebola, there's another layer on top of that the virus steals from the host cell as it leaves. But they're not cells, they are genetic parasites. So they get inside host cells and they hijack cell systems, the gene expression systems, to sort of coerce the cell to make more virus particles, and that's often at some expense to the host cell. And they come in lots of shapes and sizes, but this is Ebola virus. And even amongst the diversity of viruses that are out there, it's quite striking in appearance. I know it looks like spaghetti noodles. It's quite a simple virus. It only has 7 genes. And those 7 genes produce 7 proteins. You see them pointed out on the slide here. So just for comparison, the common chickenpox virus that a lot of us had when we were kids is way more complicated than this virus is. So it doesn't have to be fancy to be deadly. If we have a quick run-through of its structure, it's made up of an RNA genome. You might be able to see a little bit of it here. And that RNA gets wrapped up in the protein. And then outside of that is what's called the viral envelope. And you see that layer here. And that's composed of a cell membrane that it takes from the host cell as it leaves. And there's some viral proteins that are attached and inserted in that previous cellular membrane. And the rest of the proteins are involved in that hijacking process. Its target cells in the body are macrophages and dendritic cells, and it attaches to those cells and indeed enters using the surface glycoprotein. Now, macrophages and dendritic cells are front-line immune cells. They're needed to launch and direct further immune responses. So you can probably see where this is going to go. As this virus starts to take over these T cells, the host is going to be unable to mount an effective defense against it. One of the really early immune responses it interferes with is something called the type 1 interferon response. And that is the secretion of these really potent little signaling molecules called type 1 interferons. And what they do is they tell infected, even neighboring cells, to shut down key gene expression systems so that there's nothing for the virus to hijack. Right? So with this system knocked out, the virus is essentially free to replicate. And the proteins that are involved in that inhibition, you see that I've outlined them here, there's 2. And really, the coup de grace is delivered by these 2 viral proteins. There's that glycoprotein again, and another envelope protein. And these are made in large amounts as the virus is now replicating inside cells. And what those proteins do is induce macrophages to secrete another group of potent molecules that are called cytokines, pro-inflammatory cytokines. And the end result of that is that it causes the cells that line the blood vessels, the vascular system, to kind of pull apart from each other a little bit, and the blood vessels become leaky. It's a vascular permeability, and this is where the hemorrhaging part of the disease comes in. The disease itself is characterized by an incubation period of about 2 to 21 days, and that's just the time where someone is infected, but they're not actually showing any signs of illness yet. Right? And they're not contagious. For Ebola virus, they're not contagious during that period. That's not true for other viral infections, however. And then as early signs start to come on, and I like to look at these signs because it's things like fever, muscle ache, vomiting, diarrhea. Ever had those signs before? Right? And this time of year, often in Canada, we might think that that's influenza, maybe food poisoning. But in Africa, it's often signs of dysentery, which is rampant in these areas. And that's a bacterial waterborne infection. Or yellow fever, or a big one, malaria. And those are transmitted by mosquitoes. They're not contacted from person to person. And so it gets misdiagnosed, these early symptoms, and so the disease spreads until somebody figures out it's Ebola. As the disease progresses through, some people will show a skin rash. It's hard to see on darkly pigmented skin, though. Bleeding from the eyes sometimes happens, nosebleeds, bleeding from the gum, and that's all evidence of vascular damage. Most of the damage, most of the bleeding happens internally, though. So, you've all seen these horror movies where there's blood gushing from the face. That doesn't happen. Okay? It's great fodder for horror movies, but you rarely ever see actual bleeding from the surface of the body. Fluid and blood is leaking inside, though, and so some of these patients present with distended abdomens because it's all collected inside. And of course, it's got to go somewhere, so it starts to leak out orifices. In some countries in Africa that have been dealing with Ebola, call this the red diarrhea. As this leaking is happening, of course, in the circulatory system, the blood pressure drops, and that can cause physiological shock. And at the same time, there's little clots that start to form, particularly in some of the highly vascularized organs, like the liver and the spleen, and that causes cell death, organ death, and that's the cause of death in Ebola. So, now that you know a little bit about the virus and the disease, what I really want to do is tell you Ebola's story. And it's an important story to tell, I think, because it's not just a story about a virus and some people. It's a story about ecology. It's a story about disturbance. Someone once said, you've probably heard this, if you shake the trees, stuff falls out. So, let's begin. The virus is named after the Ebola River. You see it here, I drew that in. It's a little tributary that's off the Congo River in the Democratic Republic of the Congo. I'm going to call it the DRC, but it's formerly known as Zaire. But that's not where the first outbreak actually began. The first outbreak began in South Sudan, 1976, and it involved workers in a cotton factory that had been set up in a rural area surrounded by cotton fields that used to be forest. And inside this cotton factory, there were lots of bats and rats. It was infected. The virus is unknown to science at this time, and by the time this outbreak runs its course, there are 151 deaths, and it shows a 53% mortality of the people who contract the disease. 53, more than half of them died. And our map is going to get pretty crowded here, so I will post some of this data as we go on, but I'm not always going to point it out. Even before this outbreak is done, another one starts up within weeks of the first. And this one is in Zaire. If at first thought that that's a case of spread, somebody transported it into Zaire. The first case, or the index case, the first person who was recognized as having hemorrhagic disease was a mission worker. He had been doing some traveling. And as this outbreak is roaring, the samples get sent to the CDC. And the CDC looks at the virus under the electron microscope. And they're not completely taken aback, right? But, yes? Sound is off? Can you hear me up there? Just show me your hand. Okay, thank you. I'll stand here then. There's another disease that causes a hemorrhagic fever indistinguishable from that of Ebola virus. And this was known from the 1960s. And the virus that causes this similar hemorrhagic disease is called Marburg virus. Marburg virus looks a little like Ebola. So when they saw this, they thought it was Marburg virus. And to confirm that, they tested it. They used antibodies that were specific to Marburg virus. Guess what? It wasn't a match. It's not Marburg, it's something new. And so they name it Ebola after the river because they don't want to tarnish the town's name. And so I'm going to do that. It's Yambuku, is the town where the outbreak samples came from. Okay? Some really important information comes out of these first two outbreaks. First of all, now that they've got some Ebola-specific diagnostic tools in the form of antibody, they test a couple of cases, hemorrhagic disease cases from 1972, and it's confirmed that was Ebola. So it kind of begs the question, how far back do these outbreaks go? Nobody really knows. Secondly, as they start to look at the differences between these two viruses, they find out they're not the same virus. This was not a case of human spread. These two viruses are genetically different enough that they're actually considered to be two distinct species, polar bears and brown bears. Okay? The one involved, in the outbreak in Sudan was known as Sudan Ebola, and I'm going to just show it in yellow as we go through the map. The one in the DRC is known as Zaire Ebola. So what does that mean? We've got two independent outbreaks. It's not coming from people. It's coming from somewhere else. In the forest, there's a reservoir. What's the reservoir? It's likely a mammal species. No one knows what it is. Remember the bats and the rats in the cotton factory? We go a little bit further, there's another outbreak. Well, two, really. 1977, and here we are, back in the cotton factory. And the rats and the bats are still there. But no one really tests, they don't have good molecular tools for looking at these animals. And Ebola virus, the disease, does this disappearing act. So for about 15 years, you don't hear about it. And then suddenly, starting in 1994, there's a series of outbreaks. It's boom, 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 all the way to 2007. And so a pattern starts to emerge. So we're going to whip through this pretty quickly, and this is where the map's going to get really busy. Watch for the pattern. We're going to start in Gabon. In Gabon, the outbreak is involving workers in a camp. It's a gold panning camp set up in the middle of literally the jungle. Retrospective studies show, as they investigate this, that these people were eating a lot of bushmeat, whatever they could find in the forest. And that included reports that some of them found a sick gorilla and butchered it, shared it, and ate it. Not confirmation, though, that that's where the disease was coming from. Okay, so question mark. At that same time, there's an outbreak on the Ivory Coast. Notice we're in West Africa here. But this isn't a human outbreak. This is chimpanzees, dead chimps, hundreds of them in the conservation area. A grad student from Europe autopsies a dead chimpanzee. She contracts Ebola. It's confirmed. The dead chimps, including the one she autopsies, is confirmed with Ebola. We have a source. She contracted Ebola from a chimp that had Ebola. It's a source, but is it the reservoir? The reservoir species is the place where the virus exists. It's the animal or animals where the virus exists, where it lays low, when it's not causing human outbreaks. Okay? Is this the reservoir? Let's go on. And by the way, that is another new species. We've got 3 species of Ebola so far, and there's more. DRC. City this time, 200,000 people. Where did it come from? The index case was someone who was going back and forth to the forest every day. Harvesting bushmeat, is that what he was eating? He was certainly harvesting trees. Going on, Gabon. Villagers, all of the villagers who contracted Ebola shared in the consumption of a dead chimp that they found in the forest. Likely it died of Ebola. There's also reports from the local communities that there's lots of dead gorillas in the forest. Okay? Second outbreak, hunters. Reports of dead chimps in the forest. Are you seeing the pattern here? Chimps, gorillas, great apes. Is this the reservoir? Let's go on. Now we're in the Republic of the Congo, little country here, and you can see there's an outbreak in Uganda too. Again, hunting. Chimps, gorillas, and these small forest antelopes called duikers. Large die-offs of chimps and gorillas reported. Keep going on. More outbreaks. It's the same pattern. Hunting, contact with chimps and gorillas. Reports of numerous dead chimps and gorillas. These human outbreaks and deaths in chimps and gorillas are happening concurrently. Duikers, by the way, look like this. I just had to show you a picture. They're really cute little things about this big. So, these chimps and gorillas, these dead chimps and gorillas, are actually confirmed to have Ebola virus. So Ebola virus infects the great apes. Is this the reservoir? And the answer is no, it's not. It's not the reservoir because the reservoir species, from what we know from most virus infections, the reservoir species is the home, if you like, of the virus, okay? And between the reservoir species and the virus, there's a biological balance that happens, where the virus has to be maintained within that species, and that's only going to happen for the virus to be transmitted, as long as there's low levels of morbidity, sickness, and mortality. And that's not what's happening in the apes. They're getting hit really hard. They're not the reservoir, they're victims, just like humans are. But they're source of the virus. Not the reservoir, but a source. And their populations, by the way, have been decimated by Ebola. I think in the face of the human outbreaks, we sometimes forget what's been happening to these species that were already endangered. What you're seeing is a series of spillover events. A spillover event is simply when an infectious agent from a natural or wild animal reservoir moves into another species, such as humans. Okay? And as the picture depicts, if you keep putting stuff in the glass, like let's say factories and mines and a bunch of hunters, the stuff in the glass has to move out somewhere. Okay? That's obviously an oversimplification. There's a lot of epidemiological dark matter that is involved in causing that event, but you see this pattern of encroachment, of disturbance in the forest. And by the way, bushmeat is not done on just a subsistence basis for hunting. It's an industry. Okay? Tons of animals are taken in this industry and sold. Okay? So just like mining, logging, factories, it's an industry. And by the way, bats are bushmeat too. Tastes like chicken. Well, we haven't looked at, we haven't seen what the reservoir actually is yet. What is the original home of the virus, where it's evolved and where it is in these communities? Let's keep going. We're in the DRC again. There's an outbreak in Uganda as well, but the DRC, there's a massive fruit bat migration. So there's a lot of bats in the bushmeat coming in from the forests. And the index case is the village chief who's been out hunting. Okay, so what is it about bats? Remember the bats in the cotton factory too? Okay. Are bats the holy grail? Is this the reservoir? So there's been some studies about bats. There was a big one done in 2005 where they looked at not just bats, but really hundreds of animal species at the border between Congo and Gabon, because that's where all the outbreaks of the great ape deaths and the human outbreaks were taking place. Good place to look for the reservoir, it's out there somewhere. What they found was that amongst all of those animal species, and they tested thousands of animals, they found 3 species of large fruit bats that had antibodies specific to Ebola virus. What does that mean? Well, it just means that they've been exposed to the virus. Maybe these are the survivors of their own outbreaks of Ebola disease. It doesn't prove anything. The proof, the gold standard of proof, would demand then that you can isolate live virus from the bats, and that the bats we expect should be healthy bats if they're the actual true reservoir. And that's never been done. To this day, and I think efforts will intensify, Ebola virus has never been isolated from wild bats. So what's the next best thing? You can't isolate the virus itself, but we have been able to isolate genetic material from those bat species. It's fairly convincing evidence, but it's not meeting the gold standard. And by the way, if you look at those numbers, it's kind of low. Okay? Not a lot of evidence, not a lot of antibodies, not a lot of genetic material. But maybe we expect that. Maybe that's part of the biological truce between the bats and Ebola virus, that it doesn't get to really high levels so that it doesn't cause disease. And that way it's just quietly transmitted amongst the bat species. We don't know. So here we are, 7 years later in the 2014 epidemic. It actually began retrospectively in December of 2013, right on the border of Guinea, next to Liberia, where it spread. These are not individual outbreaks, independent outbreaks with different viruses. This is human spread. And of course, if you look there, Sierra Leone really didn't stand a chance. That's where it went next. It was carried from Liberia to Nigeria by a single person on a plane. And in Nigeria, that person was brought to the hospital. He collapsed. The healthcare workers went to where he had. and 8 of them died. And that was the end of that outbreak in Nigeria. And while this is happening in West Africa, and by the way, let's go back to the bat picture for a minute. You notice the range of those bats? Yeah, Central Africa and this part of West Africa. Curious. While this is happening, West Africa, where the epidemic is raging, there's an independent, concurrent outbreak happening again in the DRC. Okay? And that one's going on. So far, and I just updated this morning, it's 40 deaths. Okay? The government of the DRC says they're confident that they can get that one under control. But right now, it's still increasing. And lastly, Senegal, a single person, young man from Guinea, got across the border to go stay with his uncle, and he had Ebola virus. He didn't infect anybody else, though. In Senegal, dodged a bullet, if you like. One case, no epidemic there. So there's a few take-home messages. One of them is, first of all, that there's not going to be eradication of this virus. There won't be eradication because Ebola virus exists in some animal reservoir species or species. Might be bats, we don't know. As long as there's an animal reservoir out there, you can't expect to eradicate it unless you eradicate the animal reservoir. And that can't be done. And bats are hundreds of thousands in their populations. So I'm looking for a silver lining for all of this, of course, and I keep coming back to the gorillas and the chimps in my mind. Maybe because they are a confirmed source of Ebola, there might be a reduction in hunting in these populations. Maybe conservation efforts can finally make some way in trying to replenish or allow replenishing of these populations. And lastly, best said, I think, by David Quammen. He's a science writer. He wrote this book in 2012. To put the matter in its starkest form, human-caused ecological pressures and disruptions are bringing animal pathogens ever more into contact with human populations, while human technology and behavior are spreading those pathogens ever more widely and quickly. And by the way, recommended reading, well-researched, not just about Ebola, but the history of HIV as well. Thank you.
Thank you, Christine. Thank you. Our next speaker is Dr. Shannon Majowicz, an assistant professor in the School of Public Health and Health Systems here at the University of Waterloo. She has degrees in biomedical science, her bachelor's, and epidemiology, her master's and PhD, from the University of Guelph, where she too remembers her professors talking about new infectious diseases like Ebola. In particular, the ways that changes in biology, the environment, and human behavior come together to create opportunities for such diseases to emerge and spread. Shannon is an infectious disease epidemiologist who focuses on zoonotic diseases, which is really just a fancy way of saying that she studies the occurrence, spread, and prevention of diseases that can be transmitted between animals and people. Her particular research area is enteric diseases, those caused by E. coli and Salmonella, for example, diseases that are transmitted via consumption of food or water that has been contaminated with human or animal poop. I didn't write that. Shannon, I'm guessing you're not really talking about work over dinner most days. Okay. Prior to joining Waterloo in 2012, she was an epidemiologist with the Public Health Agency of Canada, where, among other things, she participated in the agency's response to the 2009 H1N1 influenza pandemic. Please join me in welcoming Shannon Majowicz to speak about how Ebola spreads.
Thanks very much. So maybe I'll hug this microphone as well. I feel like I'm not projecting, but we'll see what I can do. There we go. So I'm here to talk about how Ebola spreads. And when we talk about how Ebola spreads, the first thing that we need to think about is how the actual virus gets from one person to another. This is called the transmission. And Ebola is spread through direct contact with a variety of different things, most predominantly with body fluids from someone who is sick with or has died from Ebola virus. And when we talk about direct contact, what we mean is that the virus particles from someone who is sick have to actually get into your body, either through cuts on your skin, and they can be minute cuts that you might not even be aware of, or by the virus getting in contact with one of your mucous membranes, such as those soft linings of your mouth, your nose, or your eyes. And when we think about the direct contact, it can happen not only from the actual body fluids, and there's quite a list of fluids that can transmit Ebola. It's essentially anything that can come out of your body can seem to carry the virus. We have poop, my personal area of interest, blood, which often is reported on, but also things that maybe don't come to mind as frequently, like sweat, spit, semen, breast milk. You can also contract Ebola by coming into contact with objects that have been contaminated with the virus, including needles and medical equipment, or by coming into contact with infected animals, either directly with the animal and then contacting that animal's bodily fluids, or by consumption of infected meat, which we've already heard about. One of the things that we've noticed so far about Ebola, and this is all based on the evidence that we have to date, is that it doesn't appear to be transmitted through some other important pathway, which is good news. So it doesn't appear to be mosquito-borne, transmitted from a sick person to a healthy person by bites of a mosquito. It also doesn't appear to be waterborne, which is also very good news. And it doesn't appear to be airborne. But I want to just take a moment to talk about that for a second, because I think when epidemiologists talk about airborne, we mean something a little bit different than what you might think we mean. So airborne transmission, in the sense of the way that we typically talk about it, refers to germs such as measles, where the germ can be aerosolized through exhalation or coughing or sneezing, and those minute particles can remain suspended in the air, and for up to, in the case of measles, 2 hours later, someone can come by, inhale that air, come into contact with it, and catch the disease. So Ebola is not airborne in that sense, but it is airborne in the sense that, if I was to sit next to someone, never to touch them, they were sick with Ebola, and they were to cough or sneeze directly on me, and that was able to get into my body through a cut on my skin, for example, or through mucous membranes, you would be able to get sick that way. So that's just a little point of clarification on the concept of airborne disease. Another interesting thing to think about, which I will admit I didn't actually know, is that at the time of death is the time when people are the most infectious. So at this point in time, your body has been overcome by the virus. The virus has replicated so many times that the viral load is the highest it's going to be, and you're secreting the virus through this variety of different fluids. So this is an important thing to think about when we get to some of the risk factors for spread, and when we talk about the things that we need to do to help decrease transmission. In order to decrease transmission, in theory, it's quite simple. What we need to do is we need to minimize the contact between these things listed here and unprotected people. And unprotected is underlined because with proper protection, Ebola transmission can be halted quite readily. And so, of course, this begs the question, well, that sounds simple, but what the heck do we actually do? How do we decrease contact between infected individuals and unprotected people? And there's a few main steps that I want to talk about. The first is that we need to be able to find those people who are suspected to have Ebola. Maybe they're starting to show symptoms. And we want to isolate them and monitor them, and also trace anybody that they've come into contact with while they were infectious, and isolate and monitor those people as well. And Perhaps another point of clarification about the term isolation. Again, this means not, to put someone in a room, never to have contact with anybody ever. It's not that kind of isolation, solitary confinement, but isolation from individuals who don't have the proper protective equipment on. And so here in this photo, what you can see is a healthcare worker going out into a community using a thermal scanner to try and identify people with fever, to give them information about what they would do if they were to start to develop symptoms of Ebola. The second thing that people do, that we do to really try to mitigate the transmission of the virus, is proper use of protective equipment. And so thinking about how Ebola can sort of come out of a sick body in all of these different fluids, and how it can get into a healthy body through a variety of different ways, you can see that the kind of protective equipment that you need is quite extensive. So you have surgical caps and gowns, masks to prevent the virus or contaminated fluids from getting into your mouth and nose. You have eye protection. You have long-sleeved gowns that are impenetrable to fluids, and also surgical booties. And not only do you need these things to be able to go and interact with someone who's potentially sick with Ebola, but you need to be able to change those between every contact with every other individual. So it's not only enough to have access to these, you need to have sufficient quantities that you're not reusing these as you're moving from patient to patient or person to person. The third thing that needs to happen to really try to halt transmission is sterilization and disinfection of things like floors and bed frames and medical equipment. So ideally, some of the medical equipment, like needles, would be disposable and you wouldn't reuse them. But you'll have some things that aren't disposable that need to be adequately sterilized and disinfected in between use between different patients or people. And finally, for those things that can't be readily sterilized and disinfected, you need to be able to dispose of them in a way that will prevent the virus from being transmitted. And so with things like potentially contaminated bed sheets, pillows, clothing, the way to deal with them is to incinerate them. And in terms of dealing with dead bodies, which makes the news on occasion, there needs to be proper disposal by people with full protective equipment, and the disposal needs to happen in one of two ways. So either you have a body that's disinfected, bagged in a body bag, disinfected again, and bagged again, and ideally buried in an airtight, hermetically sealed coffin, or the other option is cremation. So those are the factors that impact how the virus can move from one person to another, and the things that we do to try to prevent that spread from person 1 to person 2 and so on. But another thing we think about when we think about the spread of a disease in populations are the kinds of political and environmental and social, economic, I could go on, factors that can change the risk of spread among individuals in populations. And here in this particular outbreak, one of the most important factors are the burial practices in these West African countries. And so we all have very deeply held values about what we do and what's appropriate to do when a loved one passes. And I don't have a photo to show you or to share, but in these countries, often burial practices and proper mourning involves washing the body, touching the body, kissing the body. And you can imagine how this kind of very deeply held value can intersect with the virology, where you have the viral load highest at the point of death, to create a situation that really maximizes transmission. Another factor that was at play in this outbreak is the issue of lots and lots of contact between lots and lots of people. And one of the main factors in that is crowding. So you have large urban areas, increasing urbanization from the time of the first outbreak that Christine showed you till now. You have highly mobile populations in ways that perhaps people haven't been mobile before. And this mixing and potential for contact among a variety of different individuals and a large number of individuals can really maximize the transmission of the disease. And here you see a photo of an apartment complex in Conakry, which is the capital city of Guinea, with a population of about 2 million people. Of course, thinking about the things we need to do to prevent transmission, I hope you've made this jump that one of the things that really is a risk factor for the spread is limited healthcare in the affected regions. And so this is not only the facilities, right? So we have an example of a facility here, and then of course your ideal facility here, and a photo here showing gloves and boots that have been disinfected drying in the sun. So having enough beds and enough bed sheets and enough gloves and enough boots, etc. But also having enough healthcare workers who are trained to identify the disease, particularly because it's so easily confused with other ones, having lab capacity to be able to diagnose this. So limited healthcare, both in terms of public health capacity in individual healthcare workers, but also in the facilities and the supplies that are needed to deal with this kind of outbreak. Press the wrong button. Closely related to the idea of limited healthcare is this idea of limited surge capacity. So you have a healthcare system, and it has the ability to deal with a variety of different illnesses, with a variety of different sort of numbers of people. But when an event like this happens, you have to have extra resources and extra capacity and extra healthcare workers, because all of the health problems in the population don't magically go away when a public health crisis occurs. So the ability to add additional response, additional health facilities, and that's what's shown here is a mobile health facility from Doctors Without Borders, are needed to be able to be brought into the area to add to the existing infrastructure to deal with the outbreak. And of course, the idea of limited healthcare and limited surge capacity are really just one example of an underlying larger factor, which is poverty. And here we see a very well-stocked storeroom filled with the kinds of supplies that a healthcare facility might need. And poverty impacts not only the medical resources and the human resources needed to respond, but you think about all the incineration that has to happen to deal with this medical waste. Where does that fuel come from? Just taking fuel to do the incineration take away fuel that's needed for cooking. This is a very complex issue, and poverty plays a very large role. There are also much larger, much more long-term contextual factors that need to be at least acknowledged in terms of having a role in what's going on. And so the countries that we're talking about have a history of colonialism, there's civil unrest, there, particularly in past public health responses, has been some insensitivity to the local knowledge and culture, right? So we have Western experts, parachute in, do what needs to be done. And it doesn't necessarily always respect the kind of knowledge on the ground of the people who have been living with this issue. And I think most critically, at least for me, is the lack of trust that the affected individuals might have in both the public health agencies and in the interventions that they're proposing. And this is not an issue unique to Ebola. This is an issue that is wide-reaching throughout public health. And in fact, one of the keys for success of any public health intervention is the involvement of the public and their trust in what's being proposed. And of course, linked to lack of trust is this idea of fear and uncertainty. And fear and uncertainty in this situation abound. And it's not limited to the regional affected populations. Right? So we see the same kinds of responses from individuals on the ground here talking about their fear for what's happening with the burial of these bodies, as we see from our good friend Donald Trump, who's reacting against the thought of bringing people with Ebola back to the US. And to be frank, the kinds of things that we need to do as public health to respond to this outbreak are actually very visually frightening, right? So you imagine that this is your home, and you have people in full protective gear coming in to dispose of a body. And when I try to imagine what this would be like in a more familiar circumstance, I can't even begin to fathom the kinds of emotions that people must be experiencing. So I'd like to take a moment to address one of the questions that we often hear when we think about the spread of these kinds of diseases and the responses that we're putting into place. And it's essentially, why don't we just shut down the borders? Why don't we just halt the air travel? So here you see a picture of the border between Guinea and Liberia, separated by a very, very small waterway. So the borders between these countries are very porous. There's a lot of movement of people. And can you imagine the resources that would have to be put into place to actually police and patrol all the borders? Resources, whether money, human resources, etc., that would be much better served going into some of these public health responses that we've already talked about. The other question is, why don't we restrict air travel? So there's over 58,000 flight paths. People can get around the world in less than a day. And it is a natural question, I think, to ask, well, why don't we just stop the flights coming and going from these affected regions? And there's two issues with this. And the first is that there's so much potential movement of individuals that grounding flights from affected regions, say coming into Canada, for example, won't stop the movement of people. It won't stop people going in through a third country and flying out of that country. So it's not a very effective strategy. And I think secondly, when you look at this and you can imagine the movement of people, you also need to imagine the movement of goods. So these are the same flight paths that the gloves and the disinfectants and the healthcare workers are going to take to come into the affected regions and help with the response. And in fact, what some airports are now doing, instead of trying to ground flights, is they're creating these thermal scanning areas. And you see one being set up here, and here you see hiding behind this camera is Tom Frieden, the head of the CDC, being thermally scanned when he was in Africa as part of the response. To set these up at these major points of entry and exit so that people who might be symptomatic or having signs of a fever can be identified, and so that when they do develop symptoms, they know that they need to seek care. So I'd like to end with this question when we think about spread. And of course, I have no photos to show you and no pictures to share because we don't know what the answer to this question is. We don't know if Ebola is coming to Canada. And I think when we think about this, we can think, well, what if it does? So if Ebola comes to Canada, it's likely that we won't see the same widespread and level of mortality that we've seen so far in this outbreak. We have capacity to respond in a much greater level. And we also have a heightened sensitivity. So we are on high alert, for lack of a better word. You've seen some reports in the news of individuals who have been suspected cases. There have been 3 so far in Canada, I think, who have come back and have gone into isolation and monitoring. And so we're sort of highly sensitized and on high alert to try and find these cases if they were to arise. I think in some ways more concerning is if Ebola doesn't come to Canada. We can be tempted to think then that the problem is over and it's all gone and done. And instead, I'd like to encourage you to think of this outbreak as a symptom of a bigger underlying issue, which is essentially this global inequality, and that this inequality impacts the capacity for these nations to respond and to have the infrastructure and human resources to be able to deal with these diseases as they emerge. So as you've heard already from Christine, Ebola will always be around. And it's not only Ebola. There are many other infectious diseases that evolve and emerge. And I think the investment in the infrastructure and capacity and the reduction in the global health inequality, although it will never make the headlines on its own, is something that we can't forget. Thank you.
Thank you, Shannon. Our last speaker this evening is Professor Marc Aucoin from Chemical Engineering, and he did his undergraduate degree here at the University of Waterloo in chemical engineering before doing graduate work at the École Polytechnique de Montréal, the Université de Montréal. And he joined the University of Waterloo in 2007 after spending 5 years working at the National Research Council of Canada on the production of virus vectors for gene therapy applications. So since joining the University of Waterloo, Marc has set up a group investigating the production of complex biologics, which are therapeutic proteins such as antibodies, vaccine components, such as viruses and virus-like particles in cell culture. And to date, Marc has worked directly with close to 10 different viruses, including the more widely known influenza, HIV, and herpes. And although I would argue, Marc, that you have kids and they're in daycare, so it'd be hard to really say how many viruses you've worked with over the years. And so please join me in welcoming Marc Aucoin.
Well, thank you. Thank you for all being here tonight. I think you gave me a good introduction, so you probably know why I'm involved in this lecture, but I think that at first glance, when you probably heard that I was from a Department of Chemical Engineering, you were wondering why I would be involved in this talk. And I guess partly my mission tonight is to dispel the myth that I should be working in the oil patch. In fact, I tell you that chemical engineers, what they tend to do is to work on developing chemical processes, okay? Making something available to the wider mass, so bringing products to a great quantity of people. Can you still hear me? Okay, excellent. And with the most recent news, okay, here it says that the death toll has reached 2,630. As of yesterday, I believe the death toll was 2,800, and there were massive headlines. I don't know if you've all seen these headlines that by January, 1.4 million people have been infected by the Ebola virus. Now, that's a little sensational, okay? And this is the Associated Press and Reuters that quoted the CDC that did a study that said that between 500,000 and 1.4 million people may be infected by January. But in any case, you can see that there's going to be a huge number of people that are affected, and potentially if we wanted to release some form of treatment, the question is not going to be, do we have a treatment to give them, but do we have the facilities and the manufacturing capabilities in place to actually get these treatments made so that these treatments are available to these people. So my section of the talk is on treatments. And really, treatments are currently more keep the patient hydrated. And that's the reality, because there's no accepted or no sanctioned vaccine or therapeutic out there for Ebola. Okay? And so, really, one should ask yourself that in a period when you have, or when you have been infected by the virus, okay, and you understand that, you know, this outbreak right now, they're saying 70% lethal. If you're infected, you're probably going to try anything, right? And so, you know, give me anything, just save me. They don't care what the treatment is. If you're telling them that you might be saved, well, they're probably going to try it. And that can be actually quite dangerous. And so some of the things that I'm going to talk to you about tonight are some of the ones that are actually likely to work based on some rigorous studies. To set us on the right path, though, I want to make sure that everybody understands that we've talked a lot about antibodies so far. Antibodies are different from antibiotics. Okay? Antibiotics will kill microorganisms, but they will not work against viruses. Now, when you have the flu and you go to the doctor and you say, "Can I have some antibiotics to cure the flu?" the doctor's going to say, "No, sorry. Antibiotics don't work with the flu, because antibiotics don't work against viruses." And so antibiotics don't figure prominently as a treatment against Ebola. So by the end of this lecture, what I really want you to take away from this short talk is, one, the difference between what is a vaccine and what is a therapeutic drug, because both of them are being talked about in the media. Okay, and sometimes they're using the wording interchangeably, and there is quite a difference. And I'm going to focus on three, and there's actually quite a few more treatments or experimental treatments that exist, but I'm going to focus on ZMapp, which is a Canadian-based drug, or co-research, because it's actually a cocktail that has taken the best of what they had and put it all together to make a super formula. And I'll tell you more about it in this coming slide. A second drug, or second, well, it's actually a vaccine, and it's the VSV EBO-V, okay, and that's a vaccine that started back in the early 2000s. And a third one, which has actually been given to patients already as a test, and it's ad-free EBO-Z, and I'll tell you a bit more about this as we come along too. And that's primarily based as American experimental vaccine. So in Canada, there's only one place in Canada where you can actually work with Ebola. Okay, there's only one place that's sanctioned, and that's in Winnipeg. Okay, at the National Microbiology Laboratory, and that's because they have something that's known as a level 4 containment lab. In a level 4 containment lab, or in a level 4 containment lab, you'll have people working fully gowned like this in a pressurized suit. And the idea here is that you're not going to get infected by this virus. And level 4 is really kept for the most dangerous organisms, the most dangerous pathogens. Now, if you're going to do research on developing treatment or a vaccine or a therapeutic drug, and you want to know if it works, you're going to have to eventually work with the Ebola virus. And that's why you need that type of containment. It's not that the treatments require level 4 containment. In fact, we can actually produce treatments right here at the University of Waterloo. And in fact, my lab does a lot of work that could be designated for Ebola. It's not, but it could be. But if you're going to do challenges, so if you're going to see if these vaccines and therapeutic drugs work, and you have to test it against the Ebola virus, then you have to have this type of containment. So what is the difference between a vaccine and a therapeutic drug? Well, a vaccine is something that's administered usually to healthy people, okay? And what it does is it's trying to prompt the human body to actually mount a defense that it can use in the future if it ever gets into contact with the real virus. Okay, so it prompts the body to get ready. A therapeutic drug, on the other hand, is one that's usually administered after the fact, after a person's been infected. And what you're trying to do here is you're trying to slow down the infection. You're trying to prevent it from going any further. So our body has a natural response to viral infection. Okay? I like to say that our body is primed to fight the good fight. Okay? It'll do everything that it can to make sure that you survive. And if you do survive, it's a good thing, it prepares itself for the next battle. And this is essentially the difference between innate versus adaptive immunity, and what I'm going to be talking to you about mostly is really related to adaptive immunity. The ability of our bodies to actually create proteins that can recognize something that's foreign to itself and create, like, it's like they're working in a factory building the appropriate handcuffs. Okay? And so our body is going out and working towards making these types of handcuffs that can actually lead to stopping something foreign to the body. And this is what I'm talking about, the antibody response. Our body is able to produce antibodies, and it's going to produce a lot of different types of antibodies. And all of these antibodies that are going to be made hopefully will bind to the virus. And this is obviously just a cartoon, and in fact, the antibodies are actually much smaller than the virus. Okay? When you saw the former image, and we'll see it again after, the size of the antibody is pretty much the size of one of the small proteins on the surface. So it's not nowhere near the same size. But the idea here is that the body will create a number of different antibodies, and not all antibodies will work to prevent the spread of the infection. Okay? And this is going to become critical in the next few slides when I talk to you about some of the treatments. Only some of them might become the ones that are necessary to actually stop the infection. Okay? So I chuckled a very uncomfortable chuckle when I saw this the other day. Ebola patients buying survivors' blood from black market, WHO warns. Now, at first glance, do you think this is a good thing? Probably not. There's a lot of people shaking their heads no. I'm glad that you find it somewhat disturbing. The fact is, though, in that blood from the survivors, there's potentially antibodies there that could help in stopping the progression of the infection. And in fact, the third patient to be flown back to the United States, okay, was treated in Nebraska, and he actually got blood transfusions from the first patient that was flown back to the United States because he survived that infection. So it's not clear that this third patient survived because of the blood transfusions, but the idea is not so far-fetched, okay? That there may be antibodies in the blood that can help. But one thing is, I think you really have to make sure that what you're administering is properly tested. Because if you're just taking random blood, well, whose blood are you getting? What's in that blood? Are you passing other viral diseases, right? Is HIV coming with that blood? Is hepatitis coming with that blood? You don't know. And you don't even know, really, if the appropriate antibodies are there. Okay? Or at sufficient levels. How much do you actually need? It's not clear. So this again, is a cartoon of the virus, and what you can see, those yellow things on the outer surface are the glycoproteins. The glycoproteins are one protein in the outer membrane of of the virus, and they're very, very important. And in fact, when I talked about size of the antibodies, well, the antibodies could be binding to this protein, for example, and so the antibodies are closer to that size as opposed to very much overwhelming the virus like in the other picture. So the first treatment that I want to talk about is the one that was given to the two first Americans that were infected with the virus, 2 missionaries, and they were given something called ZMapp. Okay, and ZMapp is a nice drug. It's a cocktail of 3 monoclonal antibodies. Okay, and right now we're actually producing them. Or we, with the proverbial we, it's not we at all, it's somebody else. They're producing them in tobacco plants. Okay? And in fact, making them in the tobacco plants is actually what makes them a little bit more effective than what is traditionally used to create or to produce monoclonal antibodies, which are the Chinese hamster ovary cells in cell culture. So what ZMapp is a cocktail of 3 antibodies. And what they did was they actually took 2 separate antibody drugs, both of which were cocktails, and then they basically said, okay, well, if we do a combination of all the different permutations of antibodies, can we find the best overall drug? And in fact, that's the ZMapp is the result of the best combination. And in fact, the reason why it was allowed to be administered is that some really exciting work just got published this past summer, and it's still not, it's so new that it doesn't have page numbers on the research article. It's really ahead of print. So it was released this summer, and in Nature, the journal, not released. And what they found is that even after administering it, I believe 5 days post-infection, 100% of the animals tested survived. So it's very, very promising. And this is what was administered to these first 2 patients that were flown back to the U.S. Now, it's not just a one-time administration. It's actually multiple administrations. I think it's 3 doses, 3 days apart, was the most successful way of doing it. But the reason why the 3rd patient didn't get that same treatment, the one that had to use the blood transfusions from that first patient, is because they had none left. They had none left. Okay? The reason why they're producing them in tobacco plants is because it's a scalable method of producing it. Okay, and you can have rapid generation, but it still takes about a month to actually make enough quantity. Okay? Anybody in the audience who knows about how much protein can be made, I think each person requires about 5 milligrams of antibodies. So it's a fair amount of protein that's actually required. This is the recombinant plant, or the transgenic plant that they're making them in. They actually use a bacteria to bring in the genetic information into the leaves so that the leaves can actually produce the antibodies that can go into the patients thereafter. They can be recovered later on and used in patients. And so what this does is that we actually have a concentrated formulation of the right thing. Right? When we talked about blood, I said that there was a lot of different antibodies being produced, and not all of them are working in the way that we want them to do. Right? And that's the beauty of monoclonal antibodies, is that you're actually producing a lot of one single type. One single type that will actually be targeting what you want it to target. And actually, all 3 of them target that glycoprotein. Remember a few slides back, We talked about the yellow thing. That yellow thing is the glycoprotein. All three of them will bind to a glycoprotein, okay? But they don't all target the same location on that glycoprotein. Think of the glycoprotein as my arm, okay? And the antibodies will actually bind onto my arm. My arm is now the yellow thing, right? So some of them will bind close to my shoulder, some of them will bind close to my elbow, and some of them will bind towards my wrist. The idea is that once that it's all bound, its function is reduced. Think of it as me having to go through that door, okay? And I have my fellow speakers holding on to my arm. I might have a hard time opening the door and getting out. That's the idea of the treatment that's being done with the antibiotic, with the antibodies. So that's the first story. The second story is VSV Ebola. And this is another headline that was in the news. Canada offers experimental Ebola vaccine to West Africa, 1,000 doses. Well, what is this VSV Ebola? Well, VSV is the name of another virus, the vesicular stomatitis virus. Okay? And this virus, this Curacao virus, looks like a bullet. Okay? Slightly different than what Ebola looks like, right? It doesn't look like the worm. It looks like the bullet. Good one, right? If you're going to have a therapy, make sure it looks like a bullet. Well, VSV actually, if it infects a human, actually it is usually asymptomatic. So you actually, it's almost like you never knew that you got infected by the virus. You might have a few symptoms. You might confuse it for something else, a cold or whatnot, but it's really mild. And so what they've done is that they've taken this virus that doesn't really present any danger to humans, and they replaced its glycoprotein. Okay, so it also has this surface protein on it. Well, they basically said, let's rip that one out and put the Ebola protein on it. And by doing that, they've actually created a virus, a VSV virus, that now has that protein from Ebola on its surface. But it's not Ebola. But the body may think that it is somewhat like Ebola and mount a defense against Ebola. So that later on, it can actually then fight off Ebola. Now, one person might say to me, or ask me, well, why aren't we just giving the body the glycoprotein, the Ebola glycoprotein? If you just give them the Ebola glycoprotein, you're not going to be giving the person the virus, and so there's really not going to be any problem, because remember, at the core of all viruses is its instruction manual for the cell. Right? It's genomic content. You don't put that in there, the cell doesn't know what to do. Well, I'll tell you why we can't just give the glycoprotein, because it doesn't work. You actually need the cell to produce that protein itself. And so by bringing in this VSV Ebola virus, or VSV Ebola, what you're doing is you're bringing in the instruction set to actually make that glycoprotein within the cell. And so when it is administered to the patient will take up that virus. It will start producing what it's told to produce from the genetic information within that virus. And it will produce the Ebola glycoprotein. And when it does that, then our bodies can mount a defense. This work started, or was published in 2004, but it was started really as a response because they thought that Ebola could be a bioterrorism threat. Okay? And this was an initiative to actually go toward in that direction, to try and see how we could make sure that we were prepared in case of a terror threat. It is terrorizing, but the likelihood of it being used as a terror threat is is a lot less likely than the fact that we're having an outbreak today. So that's VSV Ebola. And it was, most of the research was done here in Canada, in Winnipeg, okay, where they actually did this for the first time, where they actually put that Ebola glycoprotein on the surface of this other virus, VSV. This vaccine is not made in plants. In this case, it's actually made in cell culture, something I'm very familiar with, and made from a cell line that's derived from the African green monkey. I should say, this vaccine hasn't actually been tested yet in humans during this outbreak, but it has been administered once before. 2 years ago, I don't know if you remember, there was a news headline that there was a patient in Germany, or a scientist in Germany, that got pricked by a needle while she was working with Ebola, and then basically was, well, scared. Canada went to the rescue, flew over this vaccine, the VSV Ebola, and administered it to the patient. And there was no negative results to that, and the patient survived. It's unclear, though, if the patient was actually infected with Ebola at the time. The last story, and very quickly, is something that actually is in clinical trial right now that has been administered to humans. It's an adenovirus-based virus vaccine. Very similar to the last example, where we actually use the virus to bring information to the cells. Remember, viruses do this very well. They are very good at getting into the cell and delivering its cargo. And its cargo, it's this booklet of information known as its genomic content. And in this one, the adenovirus is based primarily on, the outside shell is primarily based on a chimpanzee virus, okay, which doesn't elicit a huge immune response when it's administered to humans, okay? But it's able to deliver its cargo, and its cargo, again, is the gene for the glycoprotein. By delivering that gene, the cell is able to make that glycoprotein and then mount defense because that protein, which is not supposed to be there, is starting to be produced inside the human body. The difference between the adenovirus-based virus vaccine and the VSV virus vaccine is that this one is replication deficient. So while the first one, actually the whole virus replicates and makes more of itself in the human body, and the human body basically clears it because it's an innocuous virus, the adenovirus one only delivers its cargo and does not replicate. Okay? It just allows the cell to produce more glycoprotein. So in summary, there is still no proven Ebola vaccines or therapeutics that exist, but there are some really promising results. Therapeutics are used to counter Ebola once infection has happened. ZMapp is a cocktail of antibodies, and it's 3 different monoclonal antibodies, all geared towards the same Ebola glycoprotein. There's multiple vaccines that are being developed, and I haven't told you all of them. This was just a few. Vaccines aim to make your body learn so that it can defend itself. And the ones that I talked to you about today really aim towards that viral glycoprotein, which is essential for actually getting the Ebola virus into the cells. So, thank you very much. I look forward to your questions.
Thank you, Marc. So what I'm going to do now, I am aware of the time. And so what I'm going to do is a couple of things. One is, while I invite the speakers to come up to the front, I would like to first put this slide up. And what this slide does is provide you with a number of sources where you can go to obtain more information about both the local and provincial responses and information about the Ebola virus. And you'll also see that there are some links to more international sources of information about the virus itself. I've also got below that a number of 2 examples of where, if you would like to be more involved in helping, and financial help is one example of that, there's 2 organizations there that are involved in addressing some of the issues surrounding Ebola on the ground. So I'm going to leave that up there. And with respect to these questions now, what I'd like to do is propose that we leave time for questions. And I will watch the time carefully to make sure that we stop at 8:45. We're going to extend it just a little bit. So I'm giving you that warning now. It will stop, the questions, at 8:45. And in the meantime, as we now begin to ask questions of our panel of speakers, my request is that you speak clearly, because this is a very large room. What will happen after you say your question is I'm going to repeat the question, because this is being recorded for those who can't be here this evening. So I'll repeat the question for everyone's benefit and for those who are watching outside of this room, and then make sure you phrase it in the form of a question as well, which makes it easier to address. So with that said, are there questions for our speakers this evening? I'll start yes at the back. So the question was, how long does the virus survive outside of the body?
I don't know that there's a direct answer to that, because it depends on the conditions. It's got a membrane around it, which is probably easily inactivated by high temperatures. It's easily inactivated by soaps, by alcohols.
Yes, in the front. Can you repeat the question? I forgot my job here for a moment there.
Well, the question was, are we being too optimistic with regards to our future and Ebola, but also how we learned from the SARS epidemic a few years ago that may help us in working with Ebola if it ever comes to Canada.
I'll take the point on this one, but so speaking even from my own experience, I think with SARS, what it allowed us to do is to see where we really needed to be better coordinated as a nation. So you think about all of the health organizations that are involved. You have family physicians, you have local public health agencies, and provincial and federal levels. The level of coordination that needs to be there, and part of that includes legal agreements and that sort of thing. So those sorts of things are now in place, and I think it also speaks to the role that the public plays in a public health response. So if you are told that you are a suspected case and please go home and stay there and don't do anything, are you going to listen? I don't know, right? So I think we're cautiously optimistic about the future. You can't predict the future, and what you saw from the most recent CDC estimate is there's a very wide range in what we think might happen. If we can do everything we know that will help control this outbreak, it could be over by January. And if we do nothing, there could be 1.4 million cases.
I think the other thing that we have to remember is that SARS was an unknown. Okay, and we do know a lot more about Ebola, which virus it is, and the diagnostics exist. And so I think we are better prepared, okay? And I think that the fact that we did go through SARS did put in place a lot of safety measures, which we've already used. And with the 3 not cases that came to Canada, we kind of relied on that kind of expertise.
Okay. I saw there was a question there, and I see the young man in the red, so we'll start with the blue. So the question was, the drug treatment example, how were the antibodies recovered from the tobacco plant?
Well, they actually crush the leaves, okay, and then they extract them through a number of different solutions, and then they usually purify it through a number of chromatography steps. And so there's a number of steps, but really it's taking those leaves and crushing them up and putting them in solutions to be able to pull them out.
Okay, young man in red. So, can you say it one more time, louder?
When was the first Ebola outbreak? Okay.
Estimated Ebola outbreak.
The first confirmed one was in 1972.
I wasn't born then.
That wasn't the question.
I was.
In black.
Oh no, I
So can you repeat the question, Marc?
Absolutely. The question was for the use of convalescent blood. So somebody who's actually been a survivor, can we not use their blood to treat people? And whether or not, is it a Western perception that we shouldn't be doing it, and should we get over it and use something that might actually work? It hasn't been proven that it has worked. Okay, so that's the first thing. And I don't see that there's a major problem, except for the fact that I think you have to do a number of rigorous tests before actually allowing that to happen, because I think that you could actually end up causing worse scenario. Because what happens if you actually have Ebola still in the blood? Right? Are you then just spreading Ebola?
So I noticed a question far right. Okay. So when the 2 infected patients were brought back through a busy international airport like that found in Atlanta, what are your thoughts about the appropriateness of this?
So I've been in the Atlanta airport, and it is a zoo, so I can understand where your question is coming from. And I think that the thing to recognize is that this was someone who we knew had Ebola, and so we could put all of those protective measures in place. And again, you know, it's not like this individual is brought back and just sort of let out at the terminal and, okay, you know, come for a checkup in a couple of days. They're taken, you know, from where they're coming from, they're being transported with sort of full precaution and then brought to a health facility. My personal reaction is I wasn't concerned at all.
Okay, I saw 1, 2, 3, so we'll start 1. How would you assess the quantity and quality of reporting of Ebola so far?
Can you clarify? Do you mean in the media? In the media?
It varies.
Yeah.
I mean, I think the print media has tended to do a more thorough job than the sort of televised and social media, simply because of the amount of space that they have to dedicate to the story. You know, and the media is a business, and their business is to sell their story. So I think, you know, like Christine said, it's quite variable.
Okay. While I take the next question, would you be willing to bring your microphones just a little bit closer? And we'll take one at the back there. So the question is, what accounts for the variability in the mortality rate?
Okay, so there's different species, and that's part of it. So the Sudan Ebola has a lower case fatality rate than the Zaire. But even within those species, there's a lot of strain variation. So the most deadly strain of Ebola is one of the Zaire strains. That was the one that you saw 90% case fatality, okay? But there's also a context there because this is Africa. In 1970s, and right up to this day, there's no mobilization for being able to accurately test that they're truly confirmed with Ebola. So when you see all those cases, they're not confirmed cases, they're probable cases and suspect cases. And that actually can change, often dilute that case fatality rate. So when you see something that says 53%, it could be 70% if you could actually pull out who actually has Ebola. Mm-hmm. So those numbers are not accurate. There's a lot of scientific uncertainty in there because in the face of an epidemic, you cannot test everybody to confirm. Yeah, good question.
Okay, so what we'll do is start here, here, and then we'll move into the middle, and I'll reach for the balcony. So, yes. Okay, was it Clomid? Clomid, whether Clomid and the nut? Garcinia cola. Any comments on that?
No, not really.
It was a good question.
I'm going to say that, I mean, Nigeria, like us, had the benefit of, they saw it coming. They were watching for it. They knew what it was when it came, because he collapsed right in the airport with those symptoms. So that was partly why it didn't become an epidemic. Mm. No, it sounds like you're talking about it some being sort of a natural anti-Ebola or antiviral drug. Yeah.
And in fact, that's a lot of the, I said that when I started my section that most of the stuff is really hydration, but they're also giving antivirals as well. And so there will be methods like that will be given to patients in order to try and increase their survival. But I to be fair, I haven't looked into it in depth to see how those mechanisms are working.
Thank you.
If at all.
Yes,
if at all.
Mm-hmm.
So the question is whether the virus is present in the primates that are being consumed without cooking, or does it survive cooking and spicing and consumption like that?
Smoking and yeah. Okay. So the index cases that we were looking at, especially in Central Africa, were largely hunters. So they're shooting it, they're carrying the meat home, it's blood everywhere and other stuff. But they're bringing it back, someone is butchering it, so they're getting it, and there's no protection. It's hands are in there, dressing it, pulling stuff out. That carcass then goes to, or bits of carcass by this point, is going to raw meat to different households, and usually being cooked up in stew. So the cook there's a transmission there through that source. Once it's cooked, no, it's completely inactivated, which may be why you're seeing outbreaks in these small villages, because that's where it's being prepared. And then once it's smoked or cooked, it's brought into the cities. I haven't seen too many big cities where there's been outbreaks. The only one in Central Africa has been Kikwit City. And they don't even think that was related to that prepared bushmeat. It was the forest worker who brought it in. So cook it, like for most viruses, and it's completely inactivated. A lot of that meat gets smoked. And as you say, spiced. Is it properly smoked? Is it properly spiced, properly cured?
Okay. Can I just see a show of hands for who is sitting here with a question? I can just see their hand. Okay. We will not get to all questions, but we'll do our best to get to a few more. But keep in mind that the speakers, good opportunity to mention, the speakers will be staying here for at least 15 minutes afterwards, and we'll be happy to talk with you more. So I'm looking, is there any on the balcony just before? I don't want to ignore the balcony before I come back to the middle. Yes? Yes? Okay, so the question was related to how quickly the virus changes over time.
So, ladies first.
Absolutely.
It's an RNA virus. It mutates quickly. Okay. In the bats, there's evidence that there's a lot of mutant versions of it out there. There's a huge diversity of this virus in bats. What we see genetically when it moves into humans, though, is that there's really only one genetic line that moves in, does the outbreak, and then goes. What it's going to do in humans, the longer it stays in there, yeah, it's certainly capable of rapid mutation. So we don't know what those mutation rates might manifest as in humans. The longer it stays in there, the higher the probability that you might get some other variant of that virus that can transmit more readily, causes less severe disease, transmits less readily. So the longer it stays in humans, the more opportunity it has to do those things as it's done in the bats. Does that answer your question?
So I think I could, oh, sorry. I'm trying to get as many questions in. Did you want a quick comment to add?
Well, I was just going to say that with respect to the current vaccine development and therapeutic drugs, if there were to be significant mutations in the glycoprotein, yeah, it wouldn't be they wouldn't be effective, and that could be very problematic. But so far, it's been pretty consistent for quite a while with respect to the glycoprotein.
And I think it also speaks to the need for these sort of more classic infection control procedures that aren't as susceptible to mutations, right? Where you have things like protective equipment and disinfection and hand washing that are going to be effective even if there is that kind of drift.
Okay. Question from you've been patient, yes. Okay, so the question was, there has been time to prepare for what is happening now, and so what has been done in the meantime?
Right, so some of that, the historical outbreaks have all been in Central Africa. This is the first time West Africa has seen human outbreaks. So it's never been there. Why would they have been prepared? Right? It's always been historically in Central Africa. So that's part of it. And underlying that is, where are the resources going to come from? When you're living in a country, I think, that's so impoverished, why would you put resources into something that might come?
Yeah, I was going to just speak to that as well. And you look at the numbers of cases that have happened in the outbreaks that had happened in the 70s, and it's a very scary and serious disease. But at the time, I mean, it wasn't as widespread as some things like malaria, which was sort of ravishes the population. So I think it's hard to justify, and this holds for everywhere. It's hard to justify the investment of resources for something that might happen, and if you've done your job well, you have nothing to show for it because then nothing happens. When you have all of these other competing interests, it's a very difficult decision.
It's somewhat amazing, actually, that the Canadian government was so proactive in the development of these therapeutics and vaccines, and just studying a virus that really is not really affecting Canadians. So there are some very influential scientists that are behind that, and really we should be really thankful for them.
Okay, what we're looking at now is a last question scenario. So keep your hand up if you are ready for that scenario. And I'm going to go with white shirt. I'm going to let you repeat the question.
Well, the question is, with the ZMapp, which was given to the two first Americans to come back, it's a relatively new drug. Will it be worthwhile to make huge amounts of it and send it over there? That's a really tough question. It's very expensive, and I don't know if we'll be able to make enough to satisfy everybody who would need it. I think the first people that will receive any of it would be the aid workers. I don't know. I think that's where the ethical dilemmas come into play, and that's not my area.
So
keep in mind that there's no evidence it actually works. It hasn't gone through proper controlled trials. It was 2 people, 3 people.
Yeah. I mean, the best evidence right now is a study on non-human primates, and the fact that we have 2 survivors in the U.S. Remember, it was also given to, I believe, a Spaniard, and he did not survive. So 2 out of 3.
With no
Can't do
control.
stats
With
on
no
that.
controls.
Well, the only last lingering question on my mind is, why don't mosquitoes transfer it with blood? Anyone else wonder about that?
Because
If it's in the fluid.
mosquitoes don't feed on dead people. The virus is at its highest titer near death. They probably, before that, aren't getting enough viral particles to efficiently transmit that from person to person.
Okay.
While people are up and walking around.
Thank you. I hope you found this evening valuable. Thank you so much for coming. Thank you to the speakers.
Thank you.
Thank you.
Thank you. You all survived.
Well, the speakers will remain at the front if you'd like to come speak to them now with those questions you didn't get to ask. Thanks again.